Hey, hi everybody. Thanks for coming. My name's Peter Innes. I'm a postdoc at University of Colorado, Boulder, and I'll be talking today about the gene regulatory landscape during ecotypic divergence of the Great Sand Dunes prairie sunflowers. And this is a project I led during my PhD, actually, also at CU Boulder in Nolan Kane's lab. So in southern Colorado, in the San Luis Valley, there's an incredible place called Great Sand Dunes National Park, which it mainly consists of this vast field of sand dunes, which are the highest dunes in North America, right at the foot of the Sangre de Cristo Mountains. It's a really unique landscape and ecosystem, and it presents numerous challenges to life, particularly plants,
You mind repeating?
because the the dunes have low soil stability, really low nutrients, for example, nitrogen, and really extreme temperatures at the sand surface and intense exposure. So the kind of the main focus of my talk today is how do species adapt to this environment? And it turns out there's only a handful of plants that have adapted to live in the sand dunes. There's a legume, a couple grass species, and then a couple species in the sunflower family, including this rush skeleton plant and the star of the show today, the prairie sunflower. In terms of animals, there's a couple interesting species like this cute kangaroo rat, an endemic tiger beetle, as well as the giant sandworm that makes field work quite treacherous. And here's a photo of me in the field. So, anyways, back to the sunflowers. This population of prairie sunflower in the dunes is distinct from the prairie sunflowers that grow at the periphery of the dunes and beyond in the San Luis Valley. So we refer to these as the dune and the non-dune ecotype. And going back like a decade or so, there were some different ideas as to where these dune sunflowers came from. And because sand dune adaptation is something that has occurred in multiple different sunflowers. So it turns out that the dune and non-dune ecotypes in the Great Sand Dunes are genetically more similar to each other than they are to other Pediolaris subspecies or to Helianthus annuus, which suggests an independent, excuse me, an independent origin of sand dune adaptation uh, in the Great Sand Dunes. And like I said, they're distinct from each other, and in general, they have very different strategies when it comes to growth and reproduction. The dune ecotype tends to be larger, more robust, especially at the seedling stage. And most notably, they have seeds that are much larger than the non-dune ecotype, on average, like twice as large, and this difference is maintained in a common garden setting. Whereas in the non-dune ecotype, um, the seeds are much smaller, but it's more beneficial to produce a lot of them. And this has been shown really beautifully by Kate Ostovic in a reciprocal transplant experiment, showing this really strong adaptive benefit of larger seeds in the dunes, seemingly to aid with seedling provisioning. So this divergence, the differentiation between these ecotypes is occurring despite some ongoing gene flow between the populations. In some cases, the dune and the non-dune populations are within like a few hundred yards of each other. And this is a really important point because it highlights the strength of natural selection in maintaining this these differences and their divergence. And so natural selection is really kind of the primary evolutionary force in this system, it seems. At the genetic level, divergence is focused and it's highest in a few regions which are known to harbor chromosomal inversions, as shown in this FST scan here, with the red blocks showing inversion regions. And this system has become kind of a classic example for the role of chromosomal inversions in adaptation because they limit recombination within the inversions, and that keeps adaptive alleles together. And in this case, those inversions have been shown to be associated with traits like seed size as well as nitrogen use. And the blue blocks there show some of those QTL regions. Okay, so in this system, we have some idea of the genetic landscape of divergence and how these inversions are important for adaptation. But we don't know how this translates to gene expression. And this is an important question because gene expression ultimately connects genotype and phenotype and the environment. And it comprises many different regulatory, like molecular mechanisms that are complex and interconnected, right? But when it comes to molecular ecology research, we tend to focus on just like one main mechanism, I would say, which is transcription. And other regulatory mechanisms beyond transcription are understudied in how they contribute to adaptation and divergence on short evolutionary timescales. And so today I'm going to be talking about transcription, but I'm also going to be talking about another regulatory mechanism called RNA splicing. And as a little splicing 101, uh, RNA splicing occurs when the the introns in a precursor mRNA, shown by these kind of thin lines, are removed and then joined back together to produce the mature messenger RNA. And this is facilitated by a dynamic um, network of proteins and non-coding RNAs called the spliceosome. What's really cool about RNA splicing is sometimes those exons and introns can be combined and spliced in different combinations. And that's called alternative splicing. So this process, alternative splicing, generates from a single gene or single precursor mRNA, multiple different transcripts or isoforms, which can potentially have different functions in the cell. This process of alternative splicing is regulated by developmental cues, and in plants, it's really important for transitions like into flowering, for instance. It can also be regulated by abiotic stress. But importantly, for this case, for this system, it can be regulated by DNA sequence variations. For instance, uh, at the junctions of exons and introns, you have what are called splice sites, which help with spliceosomal proteins recognizing the intron and the exon. And if you have a mutation at that splice site, that can lead to different patterns of splicing or expressing alternative isoforms in different proportions. And so because it has this kind of genetic component, that means it can evolve and potentially contribute to adaptation. So that's another kind of more specific question I'll be addressing, which is what role does alternative splicing have in adaptation and divergence? Okay, so our experiment was pretty straightforward, just kind of a general like RNA sequencing experiment. So we collected seeds from non-dune and dune prairie sunflower populations, and then we grew them in the greenhouse. So they were experiencing the same environment. So any differences we see in expression or alternative splicing should be due to genetic differences rather than the environment. And then we sequence their RNA, and the main methodological aspect I want to highlight here is you need some sort of alignment tool that takes your RNA sequencing reads and aligns it to a reference genome, in this case, the common sunflower genome, in a splice-aware fashion so that you can keep track of the junctions, the boundaries between the exons and introns, and therefore have an understanding of when an intron is spliced out, or when it's retained, or when exons are kind of shuffled and occur in different combinations, suggesting some variation in splicing. So this is an important step in our bioinformatic workflow. And then I did a variety of analyses with the transcriptomic data, including this differential expression, and as I mentioned, analysis of differential splicing. So the first results I'll show are just a principal component analysis of the DNA sequence variation. So SNPs in this case. And this is kind of a confirmatory result, just showing, as we know, the ecotypes are genetically distinct. But they also are distinct when we look at variation in transcript levels, so just gene expression level. Okay, and we see the same thing when we plot variation in isoform proportions, so alternative splicing. And together, this suggests that these processes, these regulatory mechanisms of transcription and alternative splicing could either be directly or indirectly under selection and could be contributing to adaptation and divergence of the sand dune sunflowers. All right, so next we looked at the landscape of divergence, um, both in terms of SNP variation and transcript levels and splicing. And this is what we see. So at the genomic or the genetic level, panel A, just looking at genetic differentiation, FST, you can see higher FST as we expect in these inversion regions, which are highlighted in red. And there are regions other than the inversions that show elevated divergence. And we think that's just because selection is so strong in this system and differences are starting to build up beyond the inversions. But these inversion regions, they're the largest, kind of highest, show the highest genetic divergence. Okay, and when it comes to expression divergence, so divergence in transcript levels in panel B, as well as splicing divergence in panel C, in this case, basically larger absolute values indicate larger difference between the ecotypes in overall transcript levels or in isoform proportions for panel C. And likewise with these splicing and expression, it's kind of noisy, but the main takeaway here is that there seems to be a lot more differences within these same inversion regions. And that is the case. So for genes inside inversions, uh, they're more than twice as likely to be differentially expressed or differentially spliced compared to genes outside the inversions. So this suggests that structural variation in the form of inversions is having a significant impact on multiple regulatory mechanisms. All right, so what are these genes doing? What are these genes that are differentially expressed and differentially spliced? What are they doing? What's their function? Um, First of all, this is just showing a Venn diagram of the number of genes in these different categories. So we found more genes that were differentially expressed compared to those that were differentially spliced, as well as a significant overlap between the categories. For genes that were differentially spliced, we find some really interesting functions that correspond to known adaptive traits. For instance, there's a functional term that's enriched for embryo development ending in seed dormancy. This is speculative, of course, but it seems like that could be related to this difference in seed size. And we also see a couple terms seemingly related to um, nitrogen usage, which, as I mentioned, is very different between the dune and the non-dune habitats. So this is an intriguing result. And likewise, when we look at differential expression, these genes have functions related to stress response, involving abscisic acid, as well as nutrient reservoir activity. This could be related to the same nitrogen limitation in the system. And so these are compelling results that kind of further emphasize the importance of expression and splicing in this system. And again, when we look at the overlap, there's this one term again that shows up for embryo development. Okay, so that's that's basically it. Um, just some main takeaways are that the Great Sand Dune sunflowers are an independent case of sand dune adaptation, and it's involved evolution of increased seed size, tolerance of low nutrient levels, and at the genetic level, large structural variants or inversions seem to be very important. And um, my results also highlight the impact of those inversions on the regulation of gene expression and alternative splicing, and kind of underline or emphasize that this mechanism of alternative splicing has kind of been underappreciated in the molecular ecology literature, and in this case seems to be another mechanism by which adaptation may be occurring. And lastly, I just want to say that the sand dunes are a truly awe-inspiring place. I'd recommend you visit them if you can, especially when the sunflowers are popping off in late summer, like August, especially. So with that, I'll just say thank you for listening, and thanks to my co-authors as well as scientists whose work I presented, um, especially these people listed here. So I think we might be taking questions at the very end, but um, yeah, that's it. So thanks.
Thank you, Peter Innes. And it is true, yes, we can take some questions. And we, there are two options. You could either unmute yourself and ask your question directly, or you can post your question in the chat and I will read it for you. I'm happy to start with the question while people, put their questions up. Um, I was curious to know, you mentioned that this was an independent evolution of the dune ecotype in sunflowers. And I know that there may be other examples within the Helianthus genus. There are also many examples kind of in the broader family. Um, and during your splicing 101, you mentioned that abiotic factors may play a role in kind of guiding splicing in addition to these DNA.
Mhm.
Um, variations. I was aware that in one other independent example of dune evolution in the genus Encelia, there was some evidence for the effects of herbivores in kind of guiding some of the adaptation to the dune ecosystem. Do you think that herbivores may play a role maybe in the system, or could they have some kind of effect on splicing?
Yeah, that's interesting. I think it's possible. The In terms of herbivory impacting splicing, I know of fewer examples of that sort of like biotic stressor regulating splicing than like compared to things like pathogens or other abiotic stressors like drought. But I will say that I think there are fewer kind of herbivores of the sunflowers in the dunes compared to like off the dunes. It's like a relatively simpler kind of ecosystem. I do think those kangaroo rats I mentioned are an interesting, like have an interesting interaction with the sunflowers when it comes to like predation of their seeds. So that might be another like kind of biotic interaction that's important. But yeah, I'd have to think more about the herbivory.
Yes, we have a question from Christoph Oberberg.
Hi, this is Christoph from Germany. So, due to technical reasons, I missed the first slide, unfortunately. So, maybe you uh, discussed already the topic, but I'm curious to know whether this dune ecotype, only evolved once in one dune, or whether in different dune habitats the same ecotype is observed.
Yeah, there's a really, this is a system that has mainly been studied by Lauren Riesberg's group, and they've shown just recently they had a paper in Nature, Ecology, and Evolution about parallel evolution of this dune ecotype in the Great Sand Dunes, so the system I showed, but also in Texas, there's a sand dune system where prairie sunflower, same species, has evolved a very similar dune ecotype. And so those are the two um, most similar cases of sand dune adaptation in the prairie sunflower.
And
But
are they similar
yeah,
in their expression patterns and also in the alternative splicing pattern?
yeah, nobody's looked at that yet. So
Okay.
as far as gene expression and alternative splicing goes, we only have these data from the Great Sand Dunes population,
Mhm.
but I would love to do a similar experiment with the Texas sand dune sunflowers.
Okay.
We have a question that was posted to the chat by Marisa. The question is, have you tested for introgression or hybridization within the annual sunflowers? And is it possible that other sand dune specialist sunflowers have introgressed with Helianthus petiolaris sensulatu, providing some of the alleles that were beneficial for sand dune establishment?
Mhm. Yeah, so I haven't done this work, but people in the Reesberg lab have. And initially, like when we didn't kind of know the origin of these sunflowers in the Great Sand Dunes, one idea was that it was the result of hybridization between Annuus and Petiolaris, because that has happened elsewhere to produce Helianthus anomalous, I think it's called, which is adapted to sand dunes. But yeah, it turns out that's not the case for these sunflowers. You know, the dune and non-dune type are more similar to each other than they are to annuus or anomalous or any others. But I do think it's possible, like that introgression was important, like in the kind of much earlier evolutionary history of this lineage. And I think when it comes to the inversions, um, the role of introgression is um, has been shown like to have brought some of these inversion haplotypes like into Pediolaris from Annuus like a long, long time ago. And yeah, I would again point people to that. There's a new paper that the Reesberg Lab has published in Nature Ecology and Evolution about parallel evolution of this dune ecotype, and they talk about some of those introgression results.
We do have another minute or so for questions. And uh, unless anyone else has a question, I can ask just a quick follow-up to that last point that you made there about the role of hybridization in potentially creating these inversions when the first when the sunflowers were first colonizing this habitat. Are there other mechanisms that you have considered for where these kind of unique inversions and mechanisms for their adaptations may have come from?
Besides introgression, you mean?
Yeah.
Yeah. I don't know if I have a great answer for that one. I think, um, yeah, I don't know. I'll have to give that one some more thought.
Yeah. Yeah, introgression sounds like
Think so.
a leading hypothesis in that case. Um, okay, well, thank you, Peter Innes. If you'd like, you can stop sharing your screen and we can go back to Brady Bunch format here. In a moment, we're going to move to our second talk. Before we do that, I'm just going to make a couple general announcements. The first is that the Tica talks that we have had in the past are available online on the International Composite Alliance YouTube. And if you're interested in going back and learning a ton about composite, you can go there. There is also the virtual composite workshop from the BSA conference a couple years ago on there. So you can binge watch the workshop and learn a ton. The other general announcement is just a reminder that on May 21st at 1 p.m. Central Time, there's going to be a general Tica community meeting. And the purpose of the meeting is to discuss a potential governance structure for Tica moving forward. So the conversation will be guided by Jen Mandel and Mauricio Bonifacino. And the idea is to talk about kind of a transparent and inclusive structure for the group going forward, specifically around the different major projects, including the Tica website, the Global Composite Database, the Capitulum Journal, and potentially the Tica Talks webinar as well. So that's just a reminder. And then if you haven't, already received the link and the details, you can follow up with Jen Mandel, who's put her email in the chat. Unless there's any other general announcements, we'll move to our second talk. Anthony, you're welcome to share your screen. You may need to request to become a co-host.
Yes, it says I don't have permission to do that. Let's see.
Okay. You should be.
Just a moment. Yeah. It's been a few minutes since I've had to do this over Zoom. Okay.
Okay. So for our second talk today, we're excited to have Anthony Melton of the University of Montevallo in Alabama, USA. And he'll be speaking about the genomics of a keystone shrub species of Western North America. You can take it away.
So thank you very much. So I'm Anthony Melton. I am an assistant professor of biology at the University of Montevallo. It's a small public liberal arts university in rural Alabama. But today, what I'm going to be talking about is research that I performed as a postdoctoral scholar at the University. Well, there we go, at Boise State University. So I was a postdoc with Sven Berkey from summer of 2020 to 2023. Where I did a variety of research focusing on genomics and drought stress response in Artemisia tridentata. And so while I was a postdoc at Boise State, I was a member of an Idaho EPSCoR program called the GEM3 program. So GEM3 is an acronym standing for Genes by Environment. And 3 M's are modeling, mechanisms, and mapping. And so this was a project that was focused on trying to study the biology of two really important taxa within Idaho ecosystems, Artemisia tridentata, big sagebrush in terrestrial ecosystems, and then red band trout in aquatic ecosystems. And so specifically for the mechanisms team that I was a part of, we used genetic analyses, genomic analyses, and phenotype data, and some other interdisciplinary methods to try to study how organisms respond to things like drought stress to help improve predictions of how different populations would be able to respond to environmental change, particularly change such as climate change. And so I just want to highlight for a second, one thing that I was a part of while I was a postdoc at Boise State was something called the Vertically Integrated Project Program, or the VIP program. So the projects that I'm going to touch on today all had VIP students contributing to them, and all the projects that I was a part of at Boise State all had really great students who were a part of this program. It gave students an opportunity to join a lab, get experience in research, and kind of work their way up a ladder, going from students learning about how to do research to having an opportunity to mentor other students as they become sort of more senior within the VIP program. Okay. So all of my research while I was at Boise State was focused on Artemisia tridentata, member of the genus Artemisia. And this is a genus that I think has a really interesting evolutionary history. So like so many clades within the Asteraceae, has a really complex evolutionary history. There's whole genome duplication, hybridization, as well as niche and habitat shifts. So Artemisia tridentata is a member of a group called the Western North American clade, which has an evolutionary history diversifying from members of Artemisia who were originally distributed in temperate Asia, migrated over land bridges, and then into Western North America, where it went from this really humid, temperate environment into a really arid habitat, where it's diversified into somewhat small clade. And we also see within this group shifts from being herbaceous annuals to woody perennials. We also see within this clade they have relatively large genomes compared to other members of at Artemisia, some of its close relatives, about twice the size of its closest Asian relatives. So if you've ever been out west in Idaho, parts of Oregon, you've definitely seen probably really large patches of big sagebrush, Artemisia tridentata. This is a really, really important species for a lot of ecosystems in Western North America, particularly the sagebrush steppe and sagebrush scrub ecosystems. They're keystone species that creates a lot of habitat that's depended on by a lot of different species. There's over 350 species that occur just within the ecosystems that are created by big sagebrush. And so this is really ecologically important. It's also an economically important species because the sagebrush ecosystems are important areas for livestock grazing. So we have open range areas out west where cattle are allowed to roam and graze on some of the smaller forbs and grasses that grow within these ecosystems. And so out of all of the species that depend on these ecosystems, some of them are sagebrush obligates, so sagebrush steppe obligates, only occurring within the habitats created by big sagebrush. So we have things like sage grouse, pygmy rabbits, and the sage thrasher. And these species, unfortunately, are imperiled due to habitat loss. A lot of the sagebrush steppe has been decimated. Over recent decades, there's been about a 50% decline due to climate change and different human activities. So not only is the keystone species of this habitat imperiled, but of course, since it's a keystone species, loss of that habitat is going to have a big downstream effect on a bunch of other species as well. So sagebrush, Artemisia tridentata, is really well adapted to grow in pretty dry environments. So during the summer, it'll experience quite a bit of drought and heat stress. So these are climate data from a site in Idaho, or in Orchard, Idaho. So for about 160 days out of the year, it'll experience fairly high temperatures and quite a bit of drought stress. For another 120 days out of the year, it'll experience quite a bit of cold stress. So it'll go from one extreme during the summer to relatively cold environment during the winter. And so this really leaves only a short window of good climate, good optimum temperature for germination and growth prior to the onset of that drought stress during the summer. And during this time, this blue line right here represents precipitation. It's not really getting a lot of rain throughout much of the range that it occurs in. Now, Artemisia tridentata once covered over a million square kilometers of Western North America, but due to human activity, expansion of big cities, and other human activities, we've seen a big decline in areas where sagebrush steppe is currently growing. So within this outline, so this is an outline showing a fairly recent survey of the sagebrush steppe in 2019, is becoming increasingly patchy within this distribution. And in these recent decades, it's declined by around 50%. So this really economically and ecologically important habitat is under quite a bit of danger due to these big declines. Now, given the importance of the species, we see a lot of effort in reseeding and trying to restore areas where sagebrush steppe has occurred. So this is focal taxa of one of the largest reseeding efforts within the US. So the Bureau of Land Management will invest billions of dollars annually into reseeding efforts, but unfortunately, the biological success is often pretty low, sometimes only around 10%. And while there is a focus on taking seeds and small plants from certain conservation areas, climate zones that should give it some sort of adaptive advantage, there's still some variation among all the different populations. And so one of the goals within our research team was to try to identify different processes, different adaptations that may help focus seed collection and help improve some of the efforts within all the restoration projects that were going on for big sagebrush. And better understanding the biology of big sagebrush is becoming increasingly important because not only have we seen the big declines within the last 50 or so years, for the last 20 or so years, Western North America has been under extremely severe mega drought, one of the most severe climatological events in around the last 1,200 years. And so this is, again, putting even more stress on an already stressed species and ecosystem. Okay, so for the projects that I'm going to talk about, we had a few specific questions that we were interested in. One we were interested in have different populations of big sagebrush evolve unique phenotypic and transcriptomic responses that would affect their ability to respond to climate change. And then what type of genomic mechanisms may be influencing drought response within big sagebrush? So the first project that I'm going to talk about started off as a master's project by Kara Moran, who is a student of Sven Berkey's and Stephen Novak's at Boise State. It was a big greenhouse experiment, had several hundred plants from different populations, different ploidy levels. Artemisia tridentata has diploid cytotypes and tetraploid cytotypes. And so it started off as a project looking at phenotypic response within these different populations, different cytotypes, to better understand the variation that we can see within Artemisia tridentata. And then once I joined the lab, I picked up with a few more phenotypic analyses and transcriptomic analyses as well. Okay, so how have different populations of Artemisia tridentata evolved in response to drought stress? So for this in particular, focused on diploid cytotypes. And we looked at a few different metrics to try to understand how they're responding to drought stress. So we use leaf fluorescence and temperature under acute drought stress to determine how efficiently they're able to still perform photosynthesis and how quickly they're sort of shutting off those pathways to protect themselves under drought stress. And then we performed RNA-seq to make comparisons between population, treatment, and tissues. And then finally, we wrapped up with a differential gene expression analysis to try to understand what types of genes are getting differentially expressed and try to understand what sort of genetic pathways may be contributing to any phenotypic differences that we see. So here we focused on two populations, both of the diploid cytotype. So we have populations in Idaho and a population in Utah. And so for all the figures that I'll be showing from here on out, we're going to have a consistent color scheme. The Idaho population is going to be in red, and the Utah population is going to be in blue. And so for Idaho, this is coming from an area where the plants are going to experience really long drought stress throughout the summer. So from early summer, like late May, early June, all the way into October, potentially even early November, these plants aren't going to experience a lot of precipitation. So they're going to have a really long, prolonged drought stress. For Utah, they're still going to experience quite a bit of drought stress through a big part of the summer, but much earlier on in the year, the drought stress for the Utah population will be offset by the North American monsoon. So these are plants that will experience about half the period of time of drought stress that our Idaho population would. And so we make these phenotypic and transcriptomic comparisons between our populations and a couple of potential possibilities for responses that we could see. As the environment changes, we could see no changes in traits, either within a population, between treatment groups, or even among populations. So in those cases, we would see no plasticity, just the traits essentially remaining the same. We could see plasticity within a population, but between treatments. So the trait would be changing in response to a changing environment. In this case, it would be an acute drought stress treatment within the greenhouse. And then we can also see big differences in response due to differences within the genetics of the population. So here we would have a genetic gene by environment interaction, which in this case, I'm going to call it a population effect since we're just looking at two populations. So going into this experiment, my colleagues and I predicted that the Idaho population, since it undergoes that really long drought stress through the summer, would outperform the Utah population under acute drought stress. So it would maintain higher photosynthetic efficiency and maintain better growth and health throughout the course of the experiment. We tested for plasticity, which is differences between the control and a drought stress group, and then also tested for population effect, so differences between our Idaho and Utah seedlings of the same treatment. Okay. So the first thing that we compared was leaf temperature. So as these plants get stressed out, they close their stomata to reduce water loss, which leads to an increase in temperature. So if we see a big spike in temperature of the leaves, we know that they are responding to the drought stress. So this was one of our signals that the drought treatment was working. And here we see plasticity for both the Idaho population and the Utah population. And so within this figure, we have these gray lines up here at the top. The gray lines are going to represent plasticity. And then the black lines, which we'll see in a moment, are going to represent a statistically significant population effect. So here we see that both seedlings of both populations were definitely responding to the drought stress, comparing the acute drought stress treatment to the control. And next up, we see a statistically significant population effect within our drought treatment. So we have our red Idaho seedlings here, drought treatment, and drought treatment here. And So in this case, we see that the seedlings from Utah are maintaining a much higher Phi2 value, which is the efficiency of photosynthesis. So how much light that's hitting the leaves is able to get used to perform photosynthesis. And so under drought stress, the Utah seedlings are able to maintain much higher rates of photosynthesis than the Idaho seedlings. And then next up is phi NPQ, which is the amount of light that gets dissipated as heat energy. So this is a way for the plant to protect itself. It's kind of like a sunblock for the plant. We see plasticity in Idaho. So under drought stress, the Idaho seedlings are going to start dissipating more light energy. And then we also see a population effect between our drought treatments. And the Utah population is actually able to dissipate a lot less of that energy. So it's still able to maintain a lot more efficiency with using incoming light to perform photosynthesis. And so overall, looking back at our prediction that the Idaho seedlings, since they experienced that much longer, prolonged period of drought stress, would perform better when evaluated by these metrics than the Idaho seedlings. Well, we actually see that as backwards. The Utah seedlings are able to maintain much higher photosynthetic rates under drought stress than the Idaho seedlings, and that the Idaho seedlings are dissipating much more light energy under drought stress than our Utah seedlings. So contrary to our initial hypothesis, it seems that the Utah population is performing better under this short-term drought stress. And so next we wanted to figure out what may be some of the genetic mechanisms that's determining these different responses that we're seeing between the Idaho seedlings and the Utah seedlings. So from our initial greenhouse experiment, we were able to get RNA sequencing for about 78 plantlets, 121 total leaf and root samples, and perform differential gene expression analyses. Okay, so for the figure on the right, the size of the circle in the Venn diagram is related to the number of differentially expressed genes within a given group. So in this case, we're looking at genes upregulated within leaf tissue. Blue is for Utah, red is for Idaho. And as you look at it, you probably may notice that there's a big difference between the size of those circles. When we first got these results, I was really surprised at how much more differential gene expression there is comparing the control to drought stress treatment between our two different populations. So for Utah, there's a much, much higher rate of differential gene expression going from a well-watered control group to a drought stress group. And we do see some overlap within sets of genes that are getting up and down regulated. So this number, the 3,124 here, are for genes unique to Utah getting up and down regulated. And we only had about 448 that were unique to Idaho, about 850 across all the different groups that were shared among both the Utah and Idaho. And so this was a result that was pretty consistent across all the comparisons for leaf and root tissue, up, down regulated. We always saw that the Utah seedlings had much bigger transcriptomic response than the seedlings from Idaho. Now, when we look at what types of genes are getting up and down regulated, there's quite a few that you would expect, things that function in drought stress, heat stress, different osmotic regulatory pathways. But when we looked at Utah, we saw that there was quite a few genes that function in epigenomic processes, epigenetic processes like nucleosome remodeling and RNA silencing. And so these were getting downregulated in the Utah seedlings, but they weren't getting downregulated in the Idaho seedlings. Now, besides the big difference between just the number of genes getting up and down regulated within Utah compared to Idaho, I was kind of surprised by this result. We did see a lot of types of genes that we would expect. But then we weren't necessarily expecting things like different epigenomic pathway type genes. So genes that would either reduce transcription or physically change the shape of chromatin to change access to genes. And so we thought that was a really interesting result, and it was something that we wanted to dig into a little bit later on. And so overall, from this first experiment that I'm talking about, we see two big different strategies, two notable strategies between our Idaho and Utah populations. So the Idaho population, we say, has a constitutive drought priming strategy. It's pretty much always ready for stress. It's evolved to kind of expect that long drought stress through the summer, while Utah is much more plastic in its response, and it'll maintain a higher photosynthetic efficiency under a short-term drought stress because it's expecting to get that relief from the North American monsoon much sooner than the Idaho population would be receiving its precipitation to break that drought stress. And so this was looking at transcriptomic responses. But one thing within the Gem3 program that we wanted was to develop a reference genome for Artemisia tridentata to help push some of this genome to phenome type research forward. And so some of my colleagues took plants from our greenhouse experiments from those populations and performed a series of experiments to try to develop in vitro lines to be able to clone these plants to get sufficient tissue for genome sequencing and to also develop lines for future genome to phenome G/E type research. And so through these experiments, the Idaho population was picked as the reference as it performed best within the suite of experiments that was performed. So it produced more biomass within its in vitro cultures, and it had much higher survival rates. And it's also nice because this was a population that was fairly close to Boise State. And so after performing PacBio sequencing and Hi-C scaffolding to assemble the Artemisia tridentata genome, we ended up with a 4.2 gigabase haploid assembly, and we had 9 pseudochromosome scale scaffolds, which is great because the chromosome count for Artemisia tridentata is 9. So we had the vast majority of the genome assembled within these 9 pseudochromosomes. We had over 43,000 genes annotated, and we also saw that this was a really heterozygous genome coming in at 2.26%, where average for plants can be around 1%. And it was also really highly repetitive, with about 78% of the assembly being some sort of repetitive element. Okay, so next we wanted to use that new reference genome to better understand what could be going on within the genome that could influence the results that we saw within the greenhouse experiment. So we had genomes from our 2 clonal lines, the one from Idaho and the one from Utah. And then we also had one from a natural population that had been published within a paper a couple of years prior to the assembly of this reference genome. And so Artemisia tridentata has a fairly complex evolutionary history. There's diploids, there's tetraploids, there's multiple origins of the tetraploids, and that's often followed by genome size reduction. And so within this complex evolutionary history, we wanted to better understand the evolutionary history of our model lines and see what type of processes may have affected our diploid populations. And so we also wanted to, of course, better understand some of the variation within these genomes to be able to use that information to plan out future genome to phenome projects. So to better understand the evolutionary history of our genomes, we wanted to look at synteny, so conservation of major structure within chromosomes, and then also look at some of the diversity of SNPs and transposable elements within those pseudochromosomes. Okay, so first we wanted to look at synteny, and here we have 2 really boring plots. We have 2 nice, perfectly straight lines. So comparing our Utah population to the Idaho reference genome, and then also our wild Idaho, our natural Idaho population genome to the Idaho reference. And what we see is that there's no major signs of any sort of chromosomal rearrangement. So we have some very comparable genomes with no major shifts in the order of content of genes and transposable elements. Then next, we wanted to look at some of the diversity within that genome, looking at SNPs and transposable elements and where they occur within the genome. What we see is that there's a lot of consistency across the genomes in the general geography of where SNP hotspots are, so areas where SNPs are more localized. And we also see a similar thing within comparing transposable elements. So they typically occur within fairly similar areas of the chromosomes across all three of our samples that we were looking at. So then next up, we wanted to look at sequence similarity and then also look at differences between enrichment of transposable elements, as transposable elements are something that's been found to be implicated quite a bit in modification of different response pathways to stresses. Okay, so this heat map is showing sequence similarity to the reference genome. So red is actually more similar to the reference, blue is more dissimilar. And both of our genomes that we were comparing to the Idaho reference were very similar, but surprisingly, our Utah genome was actually more similar to the Idaho reference than the natural Idaho population. And after a bit of digging around, we find that the natural Idaho population is actually really close to an experimental restoration site that's been being used for different sort of reseeding experiments for several decades. So I wonder if there's any potential influence from non-native genotypes. That's something that we'll have to dig into later on. So we don't have a conclusive answer on that at the moment. And in general, across the genomes, we see fairly similar overall amount of transposable elements across the genomes. But when we dig into what those different transposable elements are, we do see some pretty big differences across the genomes. So one thing that we thought was really interesting is within our Utah genome, we see big enrichment of helitron transposable elements. So this figure here is showing the amount of transposable elements within a given window of the genome. And so if you have a dot below this red line, there's more of that specific TE within the Utah genome. And if you have a dot above the red line, there's more of it in Idaho. And we see big enrichment of helitron TEs within our Utah population or within our Utah genome. And we thought this was interesting because helitron TEs are something that's been known to pick up gene fragments, move them through the genome, and it's been implicated quite a bit in modification of response pathways. And so thinking back to the differences in expression for our Utah population relative to Idaho, this is potentially one mechanism through which that big difference may have evolved. That's something that'd be really interesting in digging into later on. But at the moment, it does seem like a really good potential for mechanism within the evolution of that response pathway within Utah. And so just to wrap things up, when comparing our Idaho and Utah populations, we see big differences within the way that they respond to drought stress. We see things from transcriptomic to phenotypic differences. Overall, we see conservation of the overall architecture of the genomes within our two diploid populations, but we do see differences in transposable element enrichment, which may be something that's contributing to those transcriptomic and phenotypic differences between our Idaho and Utah populations. And of course, all this work wouldn't have been possible without really great team of researchers. So a lot of colleagues at Boise State, Idaho EPSCoR, our VIP students. It was all big effort to push all this research forward. So many thanks to these researchers who all contributed so much to the projects. Thank you. Sorry, now I have to figure out how to stop sharing.
Thank you, Anthony. Yeah, you're welcome to stop sharing, or you can leave it there on your acknowledgement slide.
Oh, there
And we can
we
take
go. I
a
know what's happening.
we can take a couple questions here. And again, if you want to ask a question, you can raise your hand, or you can unmute yourself and ask, or you can put your question in the chat. I'm happy to read it for you. I can go ahead and start with just maybe a quick question, but I was curious. You found that the Utah populations have quite a bit more plasticity than the populations in Idaho, and that this may have to do with the summer monsoon. Do you think it might have to do with the unpredictability of that source of uh of water? How the monsoon can be, have good years and bad years?
Yeah, I would be willing to guess that that has quite a bit to do with it. Like one, it may get that reprieve from the drought stress earlier on in the year, but there's is certainly that unpredictability. So it may take more of a sort of gambling evolutionary approach here to where it's just going to grow certainly when it can try to put on that biomass earlier in the year and sort of risk whether or not it's going to be a good year or not. Whereas Idaho, it's not getting a break from that summer drought stress for a while. So it certainly is going to take it's taking a more conservative approach.
Yes, I thanks for the nice talk. Maybe I have a question. Maybe I've missed the point, but you say there are different cytotypes in the two populations, and I was wondering were there also differences in the expressions among cytotypes, or they were just among those two populations you analyzed?
So my analyses just included the diploid cytotype
Yeah.
from these two populations. Didn't have any comparisons between the tetraploids from any of the other populations. So unfortunately, yeah, those weren't included within this first suite of experiments.
Thanks.
Unless there are any other questions, and feel free to follow up with our speakers for today. I'll say thank you to Anthony, uh, and thank you to Peter for these great talks. And I will stop the recording and I'll see you all very soon. Thank you for coming today.
Thank you very much for the invitation to come present. It was great to hear Peter's talk. It was fantastic. Thank you.